Welcome to Let's Learn Plants. Today, we're going to follow the incredible microscopic journey of a single photon to understand the light-dependent reactions of photosynthesis. I mean, if you've ever looked at a green leaf and wondered exactly how it captures sunlight to power literally all life on Earth, well, you are in the exact right place. We are going to decode the biological machinery that makes it all happen. Here's the roadmap for today's explainer. We'll cover 1. Light and plant pigments, 2. The photosynthetic apparatus, 3. The Z-scheme electron flow, 4. Splitting water, 5. Proton pumping and ATP synthesis, and finally, 6. Herbicides. Let's break down these complex biochemical processes into a logical, step-by-step journey perfect for your undergrad studies. Section 1. Light and plant pigments. Okay, let's dive right in by exploring the raw fuel of our entire engine here, light, and the highly specialized plant pigments responsible for actually catching it. To really understand energy capture in a plant, we have to define our energy source first. Light doesn't just wash over a leaf in some shapeless, continuous wave. It actually travels in discrete, measurable packets of energy known as quanta. Each individual particle of light, which we call a photon, carries a very specific quantum of energy. And this beautifully illustrates the dual nature of light. On one hand, light functions as a continuous wave, completely defined by its wavelength and frequency. But on the other hand, it acts like a particle. According to Planck's law, the energy of a single photon is directly proportional to its frequency. So why does this matter for a plant? Well, because plant pigments, like chlorophyll, act like specialized molecular catcher's mitts. They're designed to catch these specific, discrete particles of energy, particularly in the red and blue parts of the visible spectrum. Now, when a chlorophyll molecule successfully catches a photon, boom, it jumps from a stable ground state into a highly unstable, excited state. But it can't just hold on to that energy forever. An excited chlorophyll molecule actually has 4 distinct choices for releasing its high energy. It can release it as a glowing red light called fluorescence, it can release it as heat, it can transfer it to a neighboring molecule, or, and this is the absolute magic of photosynthesis, it can engage in photochemistry. Only photochemistry, which is the physical transfer of a high-energy electron to an acceptor molecule, actually initiates the conversion of light into chemical fuel. This brings us to the famous Emerson Enhancement Effect. So scientists in the mid-20th century noticed something super odd. If you hit a plant with just far red light, photosynthesis drops off a cliff. It's a phenomenon known as the red drop. But if you provide red and far red light at the exact same time, the rate of photosynthesis is actually greater than the sum of the rates from each light source individually. No way, right? This posed a massive biological mystery that ultimately proved the existence of 2 completely separate photosystems working in tandem. Section 2. The photosynthetic apparatus. Let's zoom in on those 2 photosystems, the physical molecular machines that are literally embedded inside the thylakoid membrane of the chloroplast. Crucially, we really need to distinguish between the roles and locations of photosystem 1 and photosystem 2. PSI sits in the stacked grana. It absorbs light at 680 nanometers and acts as a powerful oxidant that physically splits water. On the other hand, PSI sits over in the unstacked stroma lamellae, absorbs at 700 nanometers, and produces a strong reductant to make NADPH. So they are distinct machines with totally distinct jobs. But how do these reaction centers actually get the energy in the first place? Well, think of the antenna complex surrounding them as a massive, one-way energy funnel. Hundreds of pigment molecules are perfectly organized to pass excitation energy from one to another. Because each transfer loses just a tiny, tiny fraction of energy as heat, the energy gradient moves in only one direction, straight toward the reaction center. It's this incredibly efficient, irreversible funnel, ensuring that almost every single captured photon makes it to the core. Section 3. The Z-scheme electron flow. Now that we know the physical parts, let's trace the actual chemical path of our electron through the famous Z-scheme. Tracing this Z-scheme timeline, we can track the electron's linear journey. It starts when PS2 absorbs a photon, exciting an electron that is immediately stolen away by an acceptor. The electron then rides a mobile carrier called plastoquinone over to the cytochrome B6F complex. From there, another carrier, plastocyanin, delivers it to photosystem 1. Then PSI gets hit by another photon, re-energizing that tired electron, sending it to ferredoxin, and finally using it to reduce NADP+ into NADPH. It is literally a microscopic bucket brigade of electrical current. Section 4. Splitting water, the OEC. Now, to keep this continuous flow moving, PSI has to constantly replace the electrons it keeps firing down the chain. And it does this by doing something incredibly difficult, splitting water molecules. What's really fascinating here is the step-by-step breakdown of the oxygen evolving complex, or OEC. It's basically a cluster of manganese ions that progresses through 5 distinct oxidation states, which we call S states. Every single time PSI absorbs a photon and loses an electron, it pulls one from the OEC, stepping it from S0 all the way up to S4. Once it hits that S4 state, it has accumulated enough oxidizing power to violently rip 4 electrons away from 2 water molecules, releasing the very oxygen that we breathe as a byproduct. Section 5. Proton pumping and ATP synthesis. As our electrons flow down the Z-scheme, they aren't just taking a leisurely ride. They do the heavy lifting of pumping protons, which sets us up perfectly for the creation of ATP. Let's logically break down the notoriously tricky Q cycle happening right in the middle at the cytochrome B6F complex. When plastoquinoline arrives carrying electrons, it actually takes a bifurcated path. One electron continues linearly to photosystem I, but the second electron takes a circular detour within the complex. This clever recycling loop effectively doubles the number of protons pumped across the membrane from the stroma into the thylakoid lumen. So 4 protons are pumped for every 2 electrons delivered. Because of that Q cycle and the splitting of water, the inside of the thylakoid becomes highly acidic, just packed full of protons. All those pumped protons desperately want to rush back out, and they do exactly that through ATP synthase. This is a literal spinning molecular motor, consisting of a hydrophobic channel called CF0 and a catalytic head called CF1. As protons flow through it, the enzyme physically rotates, crushing ADP and phosphate together to synthesize ATP. For your university-level understanding, you're really going to want to remember this exact stoichiometry. The ATP synthase motor requires 14 protons to complete 1 full rotation, and 1 full rotation produces exactly 3 molecules of ATP. So if you divide 14 by 3, you get a precise ratio of 4.67 protons translocated per single ATP synthesized. It's just a beautifully precise mechanical ratio. Section 6, herbicides, blocking the flow. Let's wrap up our explainer today by looking at why understanding these delicate molecular machines matters profoundly in real-world agriculture, specifically when we're talking about weed control. If a plant absorbs the herbicide DCMU, which is also known as diurin, it aggressively competes for the plastoquinone binding site on photosystem 2. Imagine sticking a piece of gum into a keyhole. Plastoquinone just can't dock, meaning the electron has nowhere to go, completely stalling photosystem 2. And without that electron flow, the plant ceases oxygen evolution, makes 0 energy, and quickly dies. Conversely, the herbicide paraquat derails the completely other end of our Z-scheme at photosystem 1. Instead of just blocking the flow, paraquat actually steals the high-energy electrons directly from PSI. It then reacts with oxygen to create highly damaging cellular radicals called superoxides. These superoxides act like biological bleach, rapidly destroying the plant's cell membranes right from the inside out. So, I'll leave you with this provocative thought from our explainer today. We've seen just how precise and delicate the light reactions of photosynthesis really are. But if we fully master the mechanics of these light reactions and understand exactly how herbicides block them or uncouple their electron flow, could we one day engineer the perfect, unblockable artificial leaf? Could we literally redesign thylakoids in future crops to be completely immune to weeds and toxins, all while maximizing food production to solve global energy crises? The answers are waiting in the very molecules we just studied. Keep learning, and I'll catch you in our next explainer.